In this video, we will continue our translocation discussion, and this time we will discuss about post-translation translocation in eukaryote in slightly in depth. So, this translation, that as we have already covered it many times, it means that mRNA is converted to a polypeptide chain with the help of ribosome. And in this case, the signal sequence is less hydrophobic. Signal sequence less hydrophobic. Because of this, it escapes the SRP and does not follow the co-translation translocation. This leads to escape from SRP binding. So what happens is that the whole translation process occurs in the cytosol, and the unfolded polypeptide chain is formed in cytosol. Here what happens after that, the cytosolic chaperone that binds to this polypeptide chain and helps in translocation to the ER membrane. Cytosolic chaperones binds to unfolded polypeptide chain, this, and then helps in translocation to the ER membrane, where it interacts with not only with SEC61 but SEC62-63 complex also. So let us consider this as a ER membrane. And this is SEC61. And this is SEC63, 62-63. And here comes this is cytosolic chaperones. The function of cytosolic chaperones is to translocate this unfolded polypeptide chain to the this SEC61, SEC62-63 complex. After that, the cytosolic chaperones unbinds from this polypeptide chain and then leaves into the cytosol, and the J domain of this SEC62, J domain of SEC62, it helps in hydrolysis of ATP, hydrolysis of BIP. ATP that is bound to BIP is hydrolysis to ADP. So what happens that the ADP contains BIP. ADP binds BIP, binds to this ADP contains BIP, binds to the polypeptide chain and try to pull it into the ER membrane, and this process continue. And as soon as more polypeptide chain come inside the ER lumen, another BIP another ADP bind BIP attached to this polypeptide chain, unfolded polypeptide chain, and helps in the traverse of this unfolded polypeptide chain inside the ER lumen. And this process continue till whole unfolded polypeptide chain comes in the ER lumen. And at this moment, BIP is also attached to the unfolded polypeptide chain. So at this point, the ADP is again converted to ATP. ADP is again converted to ATP, and this caused the release of BIP from the unfolded polypeptide chain. At the same time, the unbinding pockets of the unfolded polypeptide chain, because of which the polypeptide chains are not able to fold, are also released. Now the polypeptide chain can take its native form. So what happens in the final stage is that BIP is now bind to ATP. Because of this, they are released ATP, and this polypeptide chain, unfolded polypeptide chain, will acquire its native structure based on the sequence of amino acid. Because as with the release of BIP, when ADP is converted to ATP, not only the BIP releases from the unfolded polypeptide chain, but it also unlocks, one can say unlocks the binding pockets of the unfolded polypeptide chain, so that now polypeptide chain can form its native structure. So this is the somewhat depth, not in complete depth, but somewhat in depth information about post-translation translocation in eukaryotes. Thank you for your time.